Okay, let's get back to chapter 3 or 4, I can't remember. All right, so when we left off, we were talking about synthetic peptides. So again, there's several uses for synthetic peptides. One thing we can do with synthetic peptides is we can use it to raise antibodies against the sequence of that peptide. And again, one problem with using peptides as a vaccine antigen is that their small size makes them not very antigenic. So we need ways to increase the antigenicity. Two ways to increase the antigenicity are by linking it to a carrier protein, such as KLH, or making a multiple antigenic peptide. So we can take a polylysine loop and then conjugate the peptide to the polylysine loop. Again, this is going to increase the size. We're going to have a higher molecular weight vaccine antigen, and that's going to increase the immunogenicity. Another use for synthetic peptides is to isolate receptors for many hormones and other signals. So we can essentially use these peptides to do affinity chromatography. So if we're trying to isolate another protein that has that binds a specific protein sequence, we can have that sequence synthesized as a, pardon me, we can have that sequence synthesized into a synthetic peptide, then conjugate those synthetic peptides onto, say, Sephiroth beads, some sort of a solid phase that we can use in chromatography, and then we can isolate, so we can pour like a crude lysate from the cell here. We can pour that over the column, and our protein will stick specifically to the peptides conjugated to the Sephiroth beads. Okay, so we can use them to do affinity chromatography. Peptides can be synthesized by automatic solid phase methods. Again, so we're talking about uses for these synthetic peptides. We can use them to study structure and the rules for governing the three-dimensional structures of proteins. So we already talked about folding at home, how solving the three-dimensional structure of a protein is very difficult. A secondary structure is pretty easy. I mean, we could go online now, find some online bioinformatics tools that are pretty good at predicting secondary structure, like alpha helices, beta pleated sheets, random turns and coils. However, solving the tertiary structure of a protein is a lot more challenging. And if we're going to do it using computers, you know, we need big supercomputers in order to solve these problems. Another tool that people have in the toolbox if they're trying to solve the structure of proteins is synthetic peptides. Okay, so by making synthetic peptides, that can help us to predict what type of tertiary structure that part of a protein might fold into. We can also include unusual amino acids into synthetic peptides. So we know that there are 20 amino acids, each with a unique side chain or a variable side chain. Sometimes there are post-translational modifications made to proteins that will alter the amino acids and make them into one of these less common amino acids. So we have things like 4-hydroxyproline, 5-hydroxylysine. Again, these are usually modified by post-translational modification when we find them in vivo. However, we can include these in synthetic peptides by incorporating them directly as the peptides being synthesized. Okay, I think we've talked about X-ray crystallography before. So the gold standard for solving protein structure is X-ray crystallography. No better method exists for solving protein structure. And again, we do this by first crystallizing the protein, which is challenging in and of itself. So we need a very pure protein to crystallize. Any sort of contaminants are going to prevent crystallization. And again, this takes a very long time. Sometimes it takes years just to crystallize the protein before you can do X-ray crystallography. Okay, then we shoot X-rays at it, and we either use photographic film or detectors hooked up to a computer to analyze the diffraction pattern. And from that, we can generate an electron density map and then start fitting an atomic model to that. Okay, however, not all proteins can be crystallized. Okay, membrane proteins, for example. Membrane proteins are just notoriously difficult to crystallize, and sometimes you just can't get them to crystallize at all. So how do we solve protein structure if X-ray crystallography is the gold standard and we can't crystallize the protein? Well, we can use NMR. So NMR can determine the structure of proteins in solution. This eliminates the crystallization step, which is huge. Okay, crystallization is probably the most challenging aspect of X-ray crystallography. So we can do NMR, and in NMR, we're going to get this diagonal, and off-diagonal peaks indicate atoms in close proximity. All right, now, don't ask me to analyze this. NMR has always been like black magic to me. I don't know how people can actually analyze these and solve a protein structure, but the important things are these peaks that we see off diagonal, and again, they indicate atoms in close proximity. So somehow people can use this data to generate protein models. All right, so that takes us to the end of proteins and proteomes. Anybody have any questions on anything we covered before we start the next chapter? All right, I don't see any questions from Zoom either, so. All right, so next we're going to talk about enzymes. Okay, enzymes are biological catalysts that speed up chemical reactions. Okay, so enzymes catalyze or speed up chemical reactions. Almost all enzymes are made from protein, although there are a few very important ones actually that are made from RNA. When enzymes are made from RNA, we refer to them as ribozymes. Ribo from ribonucleic acid and zyme from enzyme kind of smushed together makes the word ribozyme. Catalysis takes place at the active site, a pocket that binds enzymes, reactants, or substrates. Okay, so when, let me use the whiteboard. All right, if we look at if we just look at a generic chemical reaction. Okay, so just a little terminology. Hopefully this is review for all of you. If we look at a very generic chemical reaction, A plus B going to AB going to C. A plus B are known as the reactants. So the reactants are the chemicals that are entering the metabolic pathway. Anything that's produced in the process is called an intermediate. And finally, in this example, C, the thing we end up with is called a product. So let's say this is a simple metabolic reaction. Let's say that this first reaction is mediated or catalyzed by enzyme number 1, and the second reaction is catalyzed by enzyme number 2. So there's a special term for the reactants that are acted on by an enzyme. We refer to those as that enzyme's substrate. Okay, so in this case, A and B are the substrates for enzyme number 1, and the intermediate AB is the substrate for enzyme number 2. Okay, so the thing that's acted on by an enzyme is known as that enzyme's substrate. Right. So catalysis takes place at the active site, a pocket that binds the enzyme's reactants or substrates. Okay, active sites are a specific pocket or cleft in the enzyme that specifically binds the substrate. Okay, and we can see in this diagram here, so here's the active site. Here we can see there are two different models for enzyme substrate interaction. We have the lock and key model and the induced fit model. I think we'll talk about this later. I'll be getting ahead of the PowerPoint if I explain it right now. But you can see in both cases, the active site specifically binds the substrate. Enzymes usually catalyze a single reaction or a set of closely related reactions. For example, most proteases will also hydrolyze ester bonds in vitro. So, like I said in the previous slide, enzymes are very specific for their substrate. And generally we just have one enzyme will just act on one substrate and catalyze one particular reaction or very closely related reactions. So here's an example. So one example, we have a peptide. Proteases will hydrolyze peptide bonds. So we have a peptide plus water is going to give us it's going to cleave the protein in two. So we're going to get 2 halves of the protein over here. Ester bond, so look at the similarities between ester bonds and peptide bonds. So here is the peptide bond. Here is an ester bond. Okay, so most proteases will also hydrolyze ester bonds, being closely related, very similar to a peptide bond. Okay, but again, these reactions have to be very closely related because enzymes are very specific for their substrates. Okay, proteases differ in degree of specificity. Some enzymes are a little more promiscuous with regards to their substrates, whereas some are much more specific. So one example of this is papain versus trypsin versus thrombin. Over here we can see, so this, in A, we see the hydrolysis site for, that's for trypsin. Okay, trypsin is a protease, and it will cut proteins on the carboxyl side of a positively charged amino acid. Okay, the two positively charged amino acids are lysine or arginine. Okay, so thrombin is specific for peptide bonds on the carboxyl side of a positively charged amino acid. Thrombin is a little more specific. Okay, it will only cut on the carboxyl side of an arginine. Okay, so again, we have two proteases catalyzing the same reaction. Thrombin a little more specific, and trypsin a little looser with its specificity, in that it will cut next to any positively charged amino acid. DNA polymerase 1 is even more specific. We only get less than 1 in 1,000 misincorporated nucleotides. So DNA polymerase 1, this is involved in replicating DNA. When we replicate DNA, well, replication of DNA has to be very high fidelity, right? Because every time we misincorporate a nucleotide, that could lead to a mutation, right? So DNA polymerases need to be very specific. DNA polymerase 1, again, very specific. Only about 1 in 1,000 nucleotides is misincorporated. So again, very specific, and it has to be very specific, or else that could possibly lead to mutations that could be deleterious or even lethal. The specificity of an enzyme is due to the precise interactions of the substrate with the enzyme. This precision is the result of an intricate three-dimensional structure of the enzyme protein. So, in order excuse me, I'm trying to remember what I told my other class and what I told you guys. The interactions that specifically hold the substrate into the active site of the enzyme are weak molecular interactions. Okay, so we're talking about things like hydrogen bonding, ionic bonding, and hydrophobic interactions. Let me get a drawing tool. So we have hydrogen bonding. This isn't working. Let me go to the whiteboard. I just cannot draw with this mouse today. Okay, so again, the chemical interactions that make the active site specific for substrate and bind the substrate to the active site are weak molecular interactions like hydrogen bonding, ionic interactions, hydrophobic interactions, and van der Waals interactions. Now, it's important to note that these types of weak chemical interactions act only over very short distances. So we're talking generally between about 1.5 to 2.5 angstroms. So it's important that the active site be very complementary to the shape of the substrate in order to bring proton donors and acceptors, positive and negative charges, in close enough proximity that these weak molecular interactions can come into play. Okay, so the shape of the substrate needs to be the shape of the active site needs to be very complementary to the shape of the substrate, and so that the arrangement of proton donors, proton acceptors, positive and negative charges that can form ionic bonds or salt bridges. So the arrangement and shape needs to be very complementary. Back to the PowerPoint. Okay, so let me back up. So here we can see like enzyme substrate interactions. And again, the close complementary fit of the substrate and the active site are going to line up proton donors and acceptors that can form hydrogen bonds. They're going to line up positive and negative charges that can form ionic bonds, and also nonpolar uncharged areas that can be involved in hydrophobic interactions. Okay, so this both holds the enzyme in the active site and is responsible for the specificity of the enzyme for its substrate. Okay, many enzymes require small molecules called cofactors for activity. An enzyme without its cofactor is an apoenzyme. An enzyme with this cofactor is called a holoenzyme. There are two types of cofactors. Metals, particularly in this course when we look at active sites, particularly divalent cations we're going to see are often cofactors in the active sites of enzymes. So we have metals. Again, like I said, the divalent cations we're going to see an awful lot. So we're going to see an awful lot of zincs and magnesiums. Another type of cofactor are small organic molecules called coenzymes. Examples of coenzymes: flavin adenine dinucleotide. You might know this better by the abbreviation FAD. Of course, FAD is one of the cofactors involved in the Krebs cycle. Nicotinamide adenine dinucleotide. You might know this better as NAD. Again, NAD is one of the cofactors in glycolysis and in the Krebs cycle. Coenzyme A, you might be familiar with, again, from metabolism. Coenzyme A, acetyl-CoA. Coenzyme A is what's attached to pyruvate from glycolysis is converted into acetyl-CoA. Acetyl-CoA is the molecule that enters the Krebs cycle. So again, there's another one that you might be familiar with. Okay, so these are all examples of cofactors. Coenzymes can be either tightly or loosely bound to the enzyme. Sometimes cofactors act very much like a substrate. So they'll bind and then they'll dissociate from the enzyme. At other times, the cofactors are covalently bound to the enzyme. When they're covalently bound to the enzyme, we refer to them as prosthetic groups. So coenzymes that are tightly bound are called prosthetic groups. Loosely associated coenzymes are more like co-substrates because they do act very much like substrates. Okay, they'll bind to the enzyme, and after the reaction's over, they'll dissociate from the enzyme. An example of this is NAD when it's reduced to NADH during glycolysis. So again, it acts very much like a co-substrate. So we'll have NAD in its oxidized form. During glycolysis, it'll be reduced to NADH. And then, of course, the NADH is going to go to the electron transport chain to give up its electrons to make more ATP by oxidative phosphorylation. Enzymes transform energy from one form to another. So one example would be in photosynthesis. We have energy in the form of light, and that energy is transformed into chemical energy in the form of ATP by photosynthesis. Then we can take the chemical energy contained in ATP, and again, by catalyzing this reaction with an enzyme, we can transform the chemical energy in ATP to mechanical energy or kinetic energy by powering myosin in order to cause a cell contraction of a skeletal muscle. Okay, so enzymes transform energy from one form to the other, so we can get from light to chemical energy in the form of ATP, and then from ATP to mechanical energy in the case of a contracting muscle. Okay, so Gibbs free energy, which is abbreviated capital G, is a measure of useful energy, or the energy available to do work. A reaction can only take place spontaneously if the change in free energy, which is delta G, is negative, making the reaction an exergonic reaction. Okay, so let's look at this chart. Okay, so here we have a substrate, and there is a certain free energy associated with this substrate. Okay. In this reaction, the product has a lower free energy than the substrate. Okay. The difference in the free energy, so the difference in the free energy contained in the substrate and the product is the delta G, or the change in free energy. Okay. Now, in this case, because the free energy in the product is lower than the free energy in the substrate, delta G is going to have a negative value. Negative delta G means that we have an exergonic reaction. So in other words, exergonic is energy out. And also, when we have an exergonic reaction, this means that these reactions will tend to happen spontaneously. So a reaction cannot take place spontaneously. It can take place spontaneously only if the change in free energy is negative, exergonic. A system at equilibrium, a system is at equilibrium, and no net change can take place if delta G is 0. A reaction cannot take place spontaneously if the delta G is positive. A net input of energy is required to drive the reaction forward. So if we were to look So if we have G on the y-axis and we have the progress of the reaction on the x-axis, if we have reactants with lower free energy than the products, okay, so this is the opposite situation. We would have a positive delta G here. So these reactions will tend not to happen spontaneously, and we need some sort of an input of energy in order to drive this reaction forward. So one example is we can couple reactions to the hydrolysis of ATP in order to give it the energy to drive it forward, or we could use an energy source like photosynthesis. We can use the energy from sunlight to drive forward exergonic reactions. Okay, so the delta G, or change in free energy of a reaction, depends only on the free energy of the products, the final state, minus the free energy of the reactants, the initial state. Delta G is independent of the molecular mechanism of the transformation. The delta G for the oxidation of glucose to CO2 and H2O is the same for combustion and aerobic respiration. Okay. Let's go back to the whiteboard for a second. Okay, so using this example of the combustion of glucose, okay, so glucose is a highly reduced molecule. It has a lot of carbon-hydrogen bonds that can be stripped of electrons for energy. And we can combust glucose to carbon dioxide and water. All right, now if we were to do this, we can do this in the laboratory. So let's say we go to the laboratory, we get some glucose, put it in a test tube, hold it over a Bunsen burner. We can do this reaction. Okay, we can combust glucose into CO2 and water. Now, in order to do that in the laboratory, we're going to have to heat this glucose to over 100 degrees Celsius to combust it. Yet, that's the very same reaction that our bodies do at about 37 degrees Celsius. Okay, the difference being when we combust glucose for energy in our bodies, the reaction is broken up into a lot of little steps, and each step is catalyzed by an enzyme to lower the activation energy. Okay, if we're just going to do this in the laboratory, we're going to have a higher energy barrier. We're going to have more activation energy to overcome. But the point of this is the delta G, or the change in free energy from the combustion of glucose, is independent of how we do it. So this delta G is the same for combusting glucose in our bodies for energy as it is combusting this in a test tube in the laboratory. Okay, delta G is independent. This is exactly the same whether we're burning it in our bodies or we're burning it in a test tube. Okay, so the delta G provides no information about the rate of a reaction. The rate of the reaction depends on the free energy of activation. Free energy of activation is this value here. Okay, so while exergonic reactions tend to happen spontaneously, there is an energy barrier that we have to overcome. Okay, that energy barrier is the energy of activation. So here's the energy of activation here. These energies of activation are actually very useful for storing energy. Okay, so if one example I like to use is your car. So your car is an internal combustion engine. It combusts gasoline for energy. And there's a certain energy of activation that has to be overcome before your car can burn gasoline for energy, right? The spark plug needs to, when the spark plug provides a spark, that overcomes the energy of activation to combust the gasoline so that we can use it for energy. Okay? If we didn't have these energy barriers, then gasoline would be a whole lot more unstable. And rather than using the energy in the gasoline for internal combustion to propel the car, it would just spontaneously blow up. And that would be no good if our cars just spontaneously blew up. Okay, so these energy barriers are actually useful when it comes to storing energy. Okay, and one more thing I want to emphasize before we move on. Enzymes catalyze reactions by lowering the energy of activation. So here we see in this reaction, we have this energy barrier. The energy of activation is the energy that we have to put in to overcome that energy barrier. If we add an enzyme, so this dashed line is the energy of activation with an enzyme present. In other words, we're catalyzing or speeding up the reaction. So enzymes work by lowering the energy of activation. All right. How do they do that? Well, let's just imagine a second order reaction. So we have two reactants that are going to react together and give us a product. So just say that, so A plus B is going to give us the product C. So just say, for example, my two fists are, we have reactant A and reactant B. Now, in order for a reaction to occur, say like A and B are floating around in solution. In order for a reaction to occur, A and B have to encounter each other. And not only do they have to encounter each other, they have to encounter each other with enough kinetic energy for a reaction to take place. And not only that, they have to be held in the correct orientation. So they have to encounter each other with enough kinetic force and correctly oriented in space that a reaction will occur. Okay. Well, how can an enzyme speed that up? Well, remember, enzymes have active sites, right? The active site can bind reactants A and B, bringing them together in space close enough that a reaction can occur. Okay. When the active site of the enzyme binds A and B, it can also hold them in the correct orientation for a reaction to occur. Also, when it's holding it in the correct orientation, when an enzyme binds to its substrate, the shape of the enzyme changes a little bit, and the shape of the substrate changes as well. So the enzyme is going to distort the shape of the substrate, and the shape of the enzyme is going to be distorted a little bit too. This puts stress on existing covalent bonds in the two reactants. That makes it easier to break existing covalent bonds and to form new ones. Okay, so in this way, enzymes lower the activation energy. So you can imagine if we're talking just about two enzymes floating around, We would have to heat up the solution so that they're moving faster. That means more collisions and more chance of a reaction happening. But if an enzyme is bringing them together, holding them in the right orientation, and straining the bonds, we wouldn't need as much heat for that to happen. That makes sense? Okay, so enzymes catalyze reactions by lowering the free energy of activation. Okay, so the free energy change of a reaction is related to its equilibrium constants. So delta G with like a little degree sign is the standard free energy when A, B, C, and D are present at a concentration of 1 molar. Okay, so if we set up a reaction where we have A, B, C, and D all together in concentrations of 1 molar, the change in free energy is delta G with the little degree symbol. So delta G with the degree symbol and a prime symbol, this is the standard free energy at pH 7.0, which gives us this formula here, where R is the ideal gas constant and T is absolute temperature. So the ideal gas constant and absolute temperature, these are universal constants. Absolute temperature is 273.15 Celsius. Cannot draw with this morning. That's supposed to be 273.15 Celsius. Okay. The ideal gas constant, I can't remember exactly off the top of my head. It's like 8.3 times 10 to the 7th, I believe. Don't quote me on that. That's just approximate. Okay. So that gives us this formula here. So change in free energy equals change in free energy under standard conditions plus ideal gas constant times absolute temperature times the natural log of the concentration of products over the concentration of reactants. Okay, so change in standard free, oops, sorry about that. Change in standard free energy under standard conditions at pH 7.0, this here, can be calculated by measuring the concentrations of the reactants and products when the reaction has reached equilibrium. Okay, so let's talk a little bit about equilibrium. So when this reaction goes, let me back up one. Okay, so under standard conditions, we have 1 molar concentrations of the reactants and the products. So everything is present at a concentration of 1 molar, 1 molar, 1 molar, 1 molar, 1 molar. Okay, so we set up the reaction like this, everything 1 molar, then we let it go to equilibrium. When this reaction goes to equilibrium, let's just say, for example, at equilibrium, we have 0.5 molar of each of the products and 1.5 molar of each of the products. Okay, I'm just throwing that out as an example. Now, equilibrium doesn't mean everything stops. Okay, the reaction is still proceeding in the forward and reverse directions. It's just that at any one given time, we have half molar concentrations of reactants and 1.5 molar concentrations of products. Okay, and again, I'm just throwing out these numbers as an example. So an analogy I like to use is, say you're having a party. Okay, so say you're having a party, there's music in the living room and food and drinks in the kitchen. There's always 5 people in the living room and 3 people in the kitchen. Okay, now I'm sure we've all had parties. It's not always going to be the same people in the living room and the same people in the kitchen, right? There might be some people in the living room dancing to the music that want to go to the kitchen to eat. There might be some people in the kitchen who've already eaten and they want to go back to the living room to dance. Okay. However, when your party is at equilibrium, at any given time, there's 5 people in the living room, 3 people in the kitchen. Okay, although people are constantly going back and forth and it's not always the same people. Okay, that's what equilibrium is like. So going back, if this reaction's at equilibrium, again, the reactions are still proceeding in the forward and reverse directions. However, at any given time, the concentrations of the products and the reactants are going to be the same. That makes sense? Okay, I see people fidgeting and it looks like I'm out of time, so let's stop there. So again, remember, coming up Friday the 19th, test number 1. Wednesday, it's going to be a review session. And again, I'll have a study guide for you guys available Monday evening, the 15th. So if there's no questions from the people joining by Zoom, I'm going to stop the meeting here and I will see you guys on Friday.